Okay, so what we're going to look at now is we're carrying on from when we were just talking about making DNA fragments. So we've looked at how to make DNA fragments and we covered, and I'll quickly go over them again, really quickly, those three processes of making those DNA fragments, which are using reverse transcriptase, we also covered using restriction endonuclease enzymes. And we also covered using gene machines. That was all in the recombinant DNA video. So if at any point you're not quite sure about how we get a fragment of DNA using reverse transcriptase, restriction endonucleases, or gene machines, then I suggest you pause this video and go back and look at recombinant DNA video. Otherwise, we will carry on looking at amplifying DNA fragments. Again, we did start to look at this in the recombinant DNA video. So I'll do another quick recap of what we already know about amplifying DNA fragments, and then we're going to look at PCR in a lot more detail. So there's two ways we can amplify DNA fragments. We can either amplify them in vivo or in vitro. So when we amplify them in vivo, and in vivo meaning in live organisms, so in vivo means in living organisms. This is what we looked at again in the recombinant technology section, but we'll cover that again now. So if we want to amplify DNA fragments in vivo, we basically have to transform host cells. And there's three major steps to this process. So our first step is our DNA fragment. So we've got our DNA fragment from whichever method we've used, either restriction endonucleases, a reverse transcriptase, or a gene machine. Our DNA fragment is then inserted into a vector. So that is our first step. So this is in vivo amplifying DNA fragments. So our DNA fragment, basically our first stage is our DNA fragment is inserted into a vector. This is a quick recap. So the DNA fragment is inserted into vector DNA. So it's going to go into the vector DNA. Remember, a vector is something that can be used to transfer DNA into a cell. So vectors could be plasmids, they could be bacteriophages. So remembering that plasmids are those small circular molecules of DNA that we find in bacteria. They could be bacteriophages. And remembering that bacteriophages are viruses that infect bacteria. So our DNA fragment is inserted into a vector, into the vector DNA specifically. And it's going to be either a cell that's inside a plasmid or a bacteriophage. So our vector DNA, what's going to happen is our vector DNA is going to be cut open using the same restriction endonuclease that was used to isolate the DNA. So that's really important again. So our vector DNA is going to be, I'm going to put this bit here actually, it's going to be cut open with the same restriction endonuclease. As the fragment. Why do we do that? Remember, we do that so that our sticky ends of our vector are complementary to the sticky ends of the DNA fragment. So that is to ensure that they are complementary. So make sure we've got complementary. Ooh, we can sort of see that sticky. Make sure we've got those complementary sticky ends. So vector DNA, we cut it open, making sure we're using the same restriction endonuclease as we used to isolate our DNA fragment, making sure we've got those complementary sticky ends. And then we mix together our vector DNA and our DNA fragment. So we mix vector and fragment DNA. And we mix them together in the presence of ligase. And ligase is another enzyme. And what ligase is going to do is going to join those sticky ends of the DNA fragment to the sticky ends of the vector DNA. And that it creates a process called ligation. And that's where those sticky ends are going to join. And then finally, for this, how we insert this DNA fragment, the last little bit is that our new combination of bases is going to all be there, and we now call this recombinant DNA. So quick overview of where we are at the minute. In vivo, amplifying DNA fragments in living organisms. So this is happening in living organisms. Our DNA fragment is inserted into a vector. It's inserted into vector DNA, and a vector is anything that allows us to transfer DNA into a cell. Our vectors in these situations are going to be plasmids or bacteriophages, and we're going to cut open this vector DNA with the same restriction endonuclease as the one that we use to isolate our DNA fragment. That ensures we've got those complementary sticky ends. We're then going to mix a vector and the fragment DNA together with ligase, and that's going to go through a process called ligation. And that's where the sticky ends of the complementary, which are those complementary ends, those sticky ends of the vector DNA and our DNA fragment are going to join together. And when they join together, we call this ligation. And we have now got a piece of recombinant DNA, and that is a vector DNA and a DNA fragment joined. Now, we haven't amplified yet. This is just the first point of when we get the DNA fragment into a vector. The second stage of amplification then, zoom in a bit. Now on the second stage of amplification, the vector is transferred into the DNA fragment's host cell. So we're going to, our vector is going to transfer our DNA fragment into the host cell. So what happens here is we've got our vector that is now holds our recombinant DNA, and that is going to be used to transfer the gene into the cells, and we call these host cells. If we use a plasmid vector, so this is where we've got two different situations. So if we're going to be using a plasmid vector, if we're using a plasmid as a vector, our host cells have to be persuaded to take in the plasmid vector and its DNA. So the host cells need to be persuaded. So we persuade them in different ways. There's a few ways we can do them. So if it's host bacterial cells, we could place them into ice cold calcium chloride solution to make the cell walls more permeable. The plasmids are added and the mixture is heat shocked. So it could be, so we can use things like heat shock or electric shock. There's a few different things, but that is one example. So so this is one example. It's not the be all and end all, but you don't need to know all the ins and outs, but it's that it needs to be persuaded in some way. And one of the main ways is heat shock. So you could have a host bacterial cells and we place them into ice cold calcium chloride. So I'm just going to pop calcium chloride on here as well. We put them into this ice cold calcium chloride solution and it makes their cell walls permeable. And that's really important. We've got that permeability now to allow that plasmid vectors to come in. The plasmids are then added and the mixture is then heat shocked. And that's when we heat it to around 42 degrees for about 1 to 2 minutes. And that encourages the cells to take in the plasmids. If we are using bacteriophages, the bacteriophage will infect the host bacterium, because remember, bacteriophages are viruses that infect bacteria, so they have that infection ability. So the bacteriophage actually is able to do lots of the work itself, and the bacteriophage will infect the host bacterium. And it does this by injecting its DNA into it. Phage DNA with the target gene in it then integrates into the bacterial DNA. So then we get integration. Host cells that then take up the vectors containing the gene of interest are said to be transformed. So I'm just going to zoom out a bit. So host cells that have actually took them up will then, we then say, become transformed. So a quick overview of this second section. We've made our recombinant DNA. Our vector then transfers the DNA fragment into the host cells. If we're using a plasmid, our host cell needs to be persuaded to take in this plasmid. So we pop it into the host cells into ice cold calcium chloride. It makes the cell walls of the hosts more permeable. We then add in the plasmids, heat shock it all about 42 degrees for 1 to 2 minutes, and that encourages the cells to then take in the plasmids. Whereas if we use bacteriophages, these are able to, by themselves, because of the way that they work, infect the host bacterium by injecting its DNA into the host cell, and then there's an integration of the host cell and the bacteriophage's DNA. And then finally, once the host cell has taken up the vectors that contain the gene, we then say that they are transformed. And then our final stage of this is actually identifying transformed host cells. And we want to ID transformed, which are those cells that have taken on the vector transformed host cells. So how do we do this? Well, a good fact to remember is only about 5% of host cells will actually take up the vector. So it's important to be able to identify which cells have been transformed. So we often use something called a marker gene to identify the transformed cells. So there's a few things we can do, and we're going to go through those now. So marker genes. Marker genes can be inserted into vectors at the same time as the gene to be cloned. They're inserted into vectors at the same time as the gene. So that will then mean that any transformed host cell will contain the gene to be cloned and the marker gene. We then grow the host cells on agar plates. And when we grow them on agar plates, they divide and they replicate their DNA. And what they will do is they will create a colony of cloned cells. Transformed cells will produce colonies where all the cells contain the cloned gene and the marker gene. So the marker gene can code for antibiotic resistance, which is what we looked at yesterday. So the host cells are grown on agar plates that contain the specific antibiotic. So marker genes can code for, so I'm going to put can here, can code for antibiotic resistance. And The great thing about that is if we grow them on agar plates, these host cells that contain the specific antibiotic, only our transformed cells that have the marker gene for the antibiotic resistance will survive and will grow. Or we could have it coding for fluorescence. So when the agar plate then is placed under UV light, only our transformed cells will fluoresce. So we identify the transformed cells are allowed to grow more, and we produce lots and lots of copies of the cloned gene. And the last little bit about this, so there are 3 stages of the in vivo amplifying DNA fragments. DNA fragment is inserted into a vector. Vector transfers DNA fragments into the host cell, and then we just need to ID that transformed host cell. If we want to produce specific proteins, if you want to produce the actual proteins, not just have that DNA present, we actually need promoter and terminator regions. So I'm just going to look at that now. So if we want to make proteins, we need promoter and terminator regions. So we need promoter terminator regions. And these are specific. Promoter regions are DNA sequences that tell RNA polymerase. They tell RNA polymerase when to start producing RNA. Terminator regions, as you can probably guess from this, tell it when to stop. So promoter, tell RNA polymerase when to start, where to start, and terminator regions tell the RNA polymerase when to stop. Without the right promoter regions present, the DNA fragments won't be transcribed by the host cell and the protein then won't be made. Promoter and terminator regions may be present in the vector DNA, or they may have to be added along with the fragment. What we're going to look at now is in vitro amplification of DNA fragments, and that's actually a process called PCR, polymerase chain reaction. So when we're going through DNA sequencing, we want to know its location, we go through restriction mapping, and basically, when we want to make more copies, we can then also use polymerase chain reactions. So polymerase chain reactions, PCR, it is an in vitro, so in the lab, way of cloning copies of DNA fragments. So we don't need to use vectors or host cells in these situations. So this is outside of a living organism. In vitro means outside of a living organism. We can make millions of copies of a fragment of DNA in just a few hours using PCR. And PCR has a few steps that are then repeated over and over and over again to make lots of copies. And it's a technique that is carried out using something called a thermocycler. So what happens is we set up, when we're doing PCR, we set up a reaction mixture. And that reaction mixture includes a few important things, and we need to make sure that we're calling this a reaction mixture. So a reaction mixture is called I was saying, we make up a reaction mixture, and that contains our DNA sample or the DNA fragment, because obviously it doesn't just have to be a fragment, we can use PCR for a few different things. Well, it's always for increasing the amount of DNA that we've got, but we can use it on a piece of DNA that we're then going to break up, for like when we're talking about genetic fingerprinting, or it can just be the fragment. So the DNA sample, DNA fragment to be copied, 3 nucleotides, primers, which are our short sequences of nucleotides, and then DNA polymerase. Now, primers are the short pieces of DNA, and these are going to be complementary to the base at the start of the fragment you want. And the DNA polymerase, as you know, is an enzyme, and that creates new DNA strands. And so what will happen next is We add all them into the reaction mixture, make the reaction mixture, and then we pop that inside the thermocycler. So that's all our DNA fragments, primers, nucleotides, DNA polymerase. And we pop that into the thermocycler. In the thermocycler, it heats the mixture to 95 degrees Celsius. And we do this because it breaks the hydrogen bonds between the bases of the two DNA strands, and the two DNA strands will then separate. The next stage is that the mixture then cools, and during this time, it allows the primers to anneal to the strands, and anneal just means bind. So I'll play that again. Mixture's cooled to 55 degrees. This causes the primers to join with their complementary base pairs at the end of each DNA strand, and then our DNA polymerase attaches to the double strands. Now, what's really important here is I'm saying the DNA polymerase attaches to the double strands. It isn't starting to work at this point, it just attaches. So when it cools to 55 degrees, our primers anneal, bind to the strands, and DNA polymerase attaches. The reaction mixture is then heated again, but this time to 72 degrees, and that allows our DNA polymerase to work. So that is the optimum temperature for DNA polymerase. DNA polymerase lines up those free DNA nucleotides alongside each template strand, and specific base pairing means new complementary strands are now formed. At this point, we have now got two new copies of the fragment of DNA, and that was one cycle of PCRs complete. And each cycle takes around 2 minutes. And then what will happen, as you can see in this diagram, is then the cycle starts again. The mixture will be heated to 95 degrees, and this time all 4 strands, the 2 original and the 2 new, are used as templates. During each PCR cycle, the amount of DNA doubles. So first cycle, we have 4 strands, 4 DNA fragments, second cycle 8, third cycle 16, and so on. And it can go exponentially. So we did some of this already. Quick look at gel electrophoresis again. So restriction mapping is a process of deducing structural information about a piece of DNA using restriction enzymes and gel electrophoresis. So a quick overview again. This is where we use those restriction enzymes to cut a piece of DNA wherever it finds a specific sequence of bases. So in this example, we might use EcoRI, and that recognizes and cuts the bases sequence at GATTC, and that would be our target sequence in a restriction site. We could also use HIND3, that cuts a 7 kilobyte piece of DNA into a 6.2 kilobyte and a 0.8 kilobyte piece. This shows that the piece of DNA contains a corresponding base, sequence AAGCTT, at 0.8 kilobytes from one end. So after a piece of DNA has been cut with a restriction enzyme, the lengths of the pieces are determined using that process that we talked about in the genetic fingerprinting video called gel electrophoresis. This uses electricity to separate DNA fragments by size. We load them into the wells. The smaller DNA fragments travel further and travel to the positive end, and the heavier ones travel less far and stay closer to the bottom. So negatively charged DNA loaded into the well, moves towards the positive terminal, shorter fragments move the furthest. We can then compare the results of the bands to a sample of fragments of varying size length. So the sample cut with HIND3, as you can see here. So known fragment sizes for calibration, and you always put, when you do gel electrophoresis, you always put a DNA ladder on, we call it. And it's basically a recognition point. It's basically like a key, just to let us know, well, what each section is, the size of them. So the sample cut with HIND3 shows that we've got bands at 6.2 and at 0.8. We could also use a second enzyme, and that cuts the same section of DNA into fragments of 1.2 and 5.8. but that doesn't really help us deduce it any further. So quick overview now of the Sanger sequencing method. So the classic chain termination method, or we call it Sanger sequencing, Sanger method of DNA sequencing, relies on that PCR that we've just spoken about, and specifically modified nucleotides known as dideoxynucleotides. Dideoxynucleotides. We've got those there. DDNTPs. So on these dideoxynucleotides, the absence of the oxygen atom on carbon atom 3 means that it cannot form a phosphate bridge with the next nucleotide. It's added to a string of bases during replication, so then DNA synthesis will stop. So the DDNTP, corresponding to one of the 4 bases, is added to a test tube, along with many copies of the DNA strand being sequenced, and these copies are then made using PCR. Copies of all 4 of the normal nucleotides are also added, and these are the raw ingredients for DNA replication, which will halt every time a modified base is used. For DNA replication to happen, DNA polymerase is needed, but this will not start replication by itself. So we need to attach, like we said before, a primer, which will anneal with the half strands of DNA and start the replication process. And the polymerase and primer are added to the test tube. And this set of ingredients, DDNTP, 4 bases, template DNA, DNA polymerase, and primer, is made up in 4 batches, one with each DDNTP. So then our 4 test tubes are then put through the thermocycler to go through PCR. In each tube, DNA replication occurs, with each new copy stopping whenever a dideoxynucleotide is used instead of a normal deoxynucleotide. This results in many partial copies of the template DNA, all beginning with a primer and ending with a DDNTP. The lengths of the fragments in these test tubes are now found by using that gel electrophoresis that we've spoken about the process before. And then you would get something that looks like this using southern blotting. What I want to just go through is some uses of this recombinant DNA technology. It's great to know about it, but actually what can we use it for? So we can actually make transformed organisms. So we said transformed cells, which are cells that include that vector and DNA fragment that we wanted to get. But actually now we're going to look at transform it into an organism. So microorganisms, plants and animals can all be transformed using recombinant DNA technology, and this is called genetic engineering. So when you hear the term genetic engineering a lot, we are referring to transformed organisms. So genetic engineering equals transformed organisms. So transformed microorganisms can be used using exactly the same technology as the in vivo cloning that we spoke about at the beginning of this video and in the other video the other day. So for example, foreign DNA can be inserted into a microorganism to produce lots of useful protein. So if we did it step by step really quickly, the DNA fragment, if we were doing this into a microorganism, so I'm just going to put microorganism here. Step 1, the DNA fragment containing, let's say, the insulin that we've been using an example is located using a technique. So we get that fragment. So we locate our DNA fragment. We locate that DNA fragment. We then extract and insert into a plasmid vector. Then the plasmid containing the recombinant DNA is transferred into a bacterium. Oh. Plasmid goes into a host cell. The transformed bacteria are identified, so then we want to ID the transformed bacteria, and then we want to grow them. And then finally, the insulin that is produced from the cloned genes is extracted and purified. So it's not just transformed microorganisms, which is exactly the same as we've spoken about before. We can also transform plants. We could use plants, for example, a gene that codes for a desirable protein could be inserted into a plasmid. This plasmid is then added to a bacterium, and the bacterium is used as a vector to get the gene into the plant cell. If we add the right promoter region, then the transformed cells will be able to produce the desired protein, and we can make transformed animals too. And a gene that codes for a desirable protein can then be inserted into an early animal embryo or into the egg cells of a female. If the gene is inserted into a very early embryo, all the body cells of a resulting transformed animal will end up containing the gene. Inserting it into the egg cells means that when the female reproduces, all the cells of her offspring will contain that gene. Again, these are two things that we covered in the recombinant DNA technology video. So if you're a little bit unsure, watch that one back. And remember that promoter regions that are only activated in specific cell types can be used to control exactly which of an animal's body cells the protein is produced in. If a protein is only produced in certain cells, it can be harvested more easily. So producing the protein in the wrong cells could also damage the organism. So we're going to look at the actual benefits of this recombinant DNA technology now. So some applications of this recombinant DNA technology. We can use it to modify organisms. Like I've just said, we can make transformed organisms. We can use it as a form of selective breeding. We've used to use selective breeding, but we can actually now use recombinant DNA tech to alter the genetic makeup of organisms a lot quicker than using selective breeding. Some genetically modified organisms have already been produced. So, for example, in 2006, 252 million acres of transgenic crops were grown globally. So transformed organisms can be used in a variety of ways. And what you need to do for the exam is actually be able to interpret information about how they are used. So we'll have another look at some exam questions in another video, focused on this a bit more in detail again. So here are some examples. So in agriculture, agricultural crops can be transformed so that they give higher yields. We can also have them so that they have herbicide resistance, pest resistance, disease resistance, and crops with increased nutritional value. This means that these plants can be used to reduce the risk of famine and malnutrition, so the crops of increased nutritional value. And crops can also be transformed to have pest resistance. And these include insect-resistant crops that have been engineered to produce a bacterial toxin that kills a specific pest. And this means that we don't need to use as many pesticides on the plants. This will reduce costs and reduces any environmental problems surrounding pesticides. So one example of a crop with increased nutritional value is golden rice. And golden rice is a variety of transformed rice, and it's been engineered to contain beta carotene. And beta carotene is converted into vitamin A when it is digested. And this gene that golden rice has inside of it is from a maize plant. So it contains one gene from a maize plant and one gene from a soil bacterium. And together enable the rice to produce beta carotene. So that's where the beta carotenes come from. Rice doesn't normally produce it, but with this gene from maize plants and a gene from a soil bacterium, it can produce beta carotene. And beta carotene, as we said, is used in our bodies to produce vitamin A. And so golden rice is being used to develop to reduce vitamin A deficiencies in areas where there's a shortage of the dietary vitamin A. So, for example, places like South Asia and Africa. And vitamin A deficiencies are huge problems in these areas, and up to 500,000 children per year worldwide go blind due to vitamin A deficiency. So that's really, really important. So looking in a bit more detail at this production of golden rice, so rice plants naturally contain the gene for beta carotene. This gene is expressed in the photosynthesizing parts of the plants. However, it is not expressed in the endosperm, which is the grain, the part that we eat. So the production of beta carotene relies on the presence of several enzymes, and not all of the enzymes are naturally available in the endosperm. So in order to complete the biosynthetic pathway to restore the production of beta carotene, scientists inserted two genes one from maize plant, one from a soil bacterium into a rice's genome. We can use genetically modified bacteria. Recombinant bacteria have been engineered to produce useful proteins such as antibiotics, hormones, and enzymes. So we're just going to have a look at a few of the different things. So antibiotics, as you know, they're a drug, and some species of bacteria naturally produce antibiotics. So these bacteria can be genetically modified and they can increase the quantity and rate of antibiotic production. And this helps to reduce the cost of antibiotic production. It means we can harvest the antibiotics from these bacteria. Hormones, so insulin, which is the one I keep using as an example. Insulin is used to treat type 1 diabetes, and we used to get it from animals. Cow, horse, or pig pancreas. So for individuals who had type 1 diabetes, we used to take insulin that was harvested from cows, horses, or pigs. So the issue was this insulin wasn't human insulin, so it didn't work quite as well. And obviously there was ethical issues. Maybe people who were vegan or vegetarian or had religious views on certain animals couldn't use those either. So now that we can make insulin using recombinant DNA, we can make it on a large scale by this transgenic bacteria. And the insulin that we produce is identical to that produced in the human body. So it can be used by diabetics with no side effects and works just as well as your own insulin, if you could make it. And so that's a massive improvement. And then we can make enzymes as well. So some enzymes, and remember, enzymes are biological catalysts, they're used in lots and lots of industrial processes. And these enzymes can be produced by transformed organisms. And what's great about this is it can be produced in large quantities, so we can make them for less money and reduce costs. So an example that we've got in here is cheese. So for example, chymosin or rennin is an enzyme that we use in cheese making. It used to be made from rennet, which is a substance produced in the stomach of cows, but we can now produce it by transformed organisms. This means we can now make it in large quantities, it's really cheap, and it means that we aren't having to kill any cows, so it makes some cheese suitable for vegetarians. We can also use enzymes to include lipases to use in cheese production. That can also improve the flavour, and we can also use amylase in beer production to break down starch. And looking at drugs again in a bit more detail, transgenic animals and plants can be modified to produce useful pharmaceuticals. When we do this, we call it farming with pH. So alpha-1 antitrypsin protects the lungs from damage during infection, and we can get transgenic sheep and they can produce can be produced and they contain the gene for this protein. And the gene can be pre-programmed to be expressed only in the mammary gland cells and then is secreted in the milk. And then it can be extracted and purified and used for treating emphysema sufferers. So we're not causing any harm to the sheep, and we're not having to kill them, and we're just extracting it from the milk. And then we can have xenotransplantation, and that's the transfer of organs or tissues into humans from other species, such as pigs. So transplanted organs are often rejected by the new host's immune system, as antigens on the cell surface will be recognized as foreign. If we use recombinant DNA technology, we can engineer pigs that lack an enzyme that is thought to contribute to transplant rejection, which means that the transplants are less likely to be rejected. And it's thought that actually with more development, xenotransplantation may be able to provide a large number of organ transplants, especially for individuals who are on waiting lists and are waiting a really long time. So we used to use pigs' hearts, but we had major rejection issues, more than we do with like taking it from another human. But the human waiting list is so huge. So we are now actually looking back at animals, and we use pigs because their hearts are a very similar shape, size to ours. In the exam, though, you are often asked about concerns around GM organisms. So there's lots of scientific, moral, and ethical issues surrounding the use of genetically modified organisms. So when we're looking at crop plants and agriculture, there's a lot of issues that could come up. First of all, recombinant genes could be passed from a transgenic plants to another species. So, for example, cross-pollination of crop plants, and this could cause a herbicide resistance gene to be incorporated into a plant considered a weed. And we could end up with basically a superweed. And this happens when we have that interbreeding of these wild plants and these transgenic plants. And because of that, we could then end up with an uncontrolled spread of recombinant DNA, and we don't really know the consequences of it, but we've then created these superweeds. Insect-resistant plants may be toxic to non-target organisms, and that's something that's really important to remember. So they're insect resistant, but if it gets taken on by a different organism, it could actually kill them. Because genetically modified organisms are being developed by multinational companies, there's actually a massive concern that these companies could dominate global food production, affecting local food producers, stopping farming, reducing costs, fees are paid to farmers, controlling consumers, how much you charge for food. There's a concern as well that the introduction of genetically modified herbicide-tolerant crops will cause farmers to use herbicides more widely, and this could reduce the diversity number of wild plants. And new allergens could actually be accidentally created, and known allergens could be transferred into other foods. Another issue with this is monoculture, and that's when farmers might plant only one type of transformed crop. This could make the whole crop really vulnerable to the same disease, because the plants are genetically identical. Environmentalists are also really concerned about this because monoculture reduces biodiversity, and this could damage the environment. There's also an issue with organic farmers, because organic farmers can have their crops contaminated by windblown seeds from nearby genetically modified crops. This means then that organic farmers can't sell their crops as organic, and they can lose income. With genetically modified animals, the large majority of experiments to produce genetically modified animals fail. There's lots of deaths occurring at embryo stage, as well as just before and after birth. There's also thought to be great incidence of illness in later life. Genetically modified animals also there's a high risk of abnormalities in subsequent generations. Some people believe it's unethical to allow experimental animals to suffer in order for us to develop the ideal genetically modified animal, and some animal welfare groups argue that the use of animals for all genetic manipulation is unethical. So yeah, when we were on the crop plants, we have that thing about globalization. So it goes into that industry idea here. So there's a group groups, and they're called anti-globalization activists, and they oppose this globalization. And that's basically growth of large multinational companies at the expense of smaller ones. And the theory is a few large biotech companies control some forms of genetic engineering. And as the use of this technology increases, the company gets bigger, the company gets more powerful, and this may force smaller companies out of business, making it harder for them to compete. There's also worries about labeling of the foods from animals and plants. Without proper labeling, some people think they won't have a choice about whether they're consuming food made using genetically engineered organisms or not. And some consumer markets, such as the EU, won't import genetically modified foods and products. And this can cause an economic loss to producers who have traditionally sold these. Microorganisms, if not properly contained genetically modified bacteria, could transfer recombinant DNA to other bacteria, and we don't know the effects of this. And antibiotic resistant markers are often added to genetically modified bacteria. This could cause problems if these genes spread to harmful bacteria, producing antibiotic resistant pathogens. In medicine, companies who own genetic engineering technologies may limit the use of technologies that could be saving lives. And some people worry this technology could be used unethically, so for things like designer babies. And we're just going to have a quick look at some other views of people about recombinant DNA. And the reason I'm going to go over these a few times is these questions come up all the time on the exams. And there's also an ownership issue with recombinant DNA technology. So there's also arguments about who owns genetic material once it's been removed from the human body. So if someone took my DNA, do I own it because I was the donor, or does the researcher who took it from me now own it? Some people argue that I would still hold the right because it's my genetic information. Others argue that value is created by the researcher who used it to develop a medicine or in diagnosis, so they now own it. So there's lots of arguments there. And so a small number of large corporations also own patents to particular seeds. And that means they can charge really, really high prices and sometimes include something called a technology fee. And they can require farmers to repurchase these seeds every year. So if non-GM crops are contaminated by GM crops, farmers can also be sued for breaching the patent law. So some examples here. An environmentalist might say, for it, research suggests that transgenic organisms do not survive for long in the natural environment, and any transfers of a gene would be acted against by natural selection. Against it, the genes from transgenic organisms could escape and be transferred to other organisms, damaging ecosystems and creating superweeds. A doctor, also for it, some people would argue agricultural crops could be produced to help reduce the risk of famine and malnutrition. A doctor for it, DNA and proteins of transgenic organisms are digested in the human gut, so they're unlikely to cause problems. A doctor also might be for GM because the medicines that we can make using GM could be produced more cheaply, so they're more affordable. And we could also use those transformed crops to make useful pharmaceutical products, which could make drugs available to more people in areas where refrigeration isn't available. But against it, it's impossible for us to be certain about the long-term effects on humans' health of the genes and proteins in GM foods. Animal rights activists, transfers of genes between organisms has been proved to be a natural phenomenon. Transgenic animals are very precious. Against moving genes between species is an infringement of the right of every species to exist independently. These techniques can cause suffering in animals. Transgenic charity worker, it will reduce the cost of food production, improve quality of food, humanitarian patents ensure the poorest do not suffer. But we could argue that these companies could monopolize the market, forcing up the prices. Finally, the last little bit we're going to look at is gene therapy. Recombinant DNA technology can also be used to treat human disease, and this is known as gene therapy. And gene therapy is potential treatment for genetic diseases, and it involves altering the genotype. The cause of the disease is targeted rather than just the symptoms. So Gene therapy isn't being widely used yet, but there is a form of somatic gene therapy available, and other treatments are undergoing clinical trials. So that's really important, just keep in mind. So how it works. Gene therapy involves altering the defective genes, so those mutated alleles inside cells to treat the genetic disorders and cancers. How we do it depends on whether the disorder is caused by a mutated dominant allele or two mutated recessive alleles. So we can either replace the defective gene with a normal allele, or we can supplement the gene by adding copies of the normal allele, which must be dominant to mask the effects of the defective recessive allele. So I'll explain which one we use for which. If it's caused by two recessive alleles, you can add a working dominant allele to make up for them, and that is supplementing. So that is when it has been caused by two, and this I'm going to write the cause of it here, caused by two recessive alleles. And that is when we use supplementing. So in supplementing, we add a working dominant allele, as you can see here, we're adding it in here to make up for the mutated recessive alleles. So we supplement the faulty ones, and because it's dominant, that is what will work. If it's caused by a mutated dominant allele, so this is going to be our mutated dominant allele. This is where we're going to replace. We're going to silence that dominant allele by sticking a bit of DNA in the middle of the allele so that it doesn't work anymore. Both these processes involve inserting a DNA fragment into the person's original DNA. So how you get the new allele inside the cell? Well, the allele is just inserted into cells using vectors like we explained earlier. So just like in recombinant DNA technology, and we can use different vectors. It can be altered viruses, plasmids, or liposomes. And then there is two types of gene therapy, somatic and germline, which we'll look at in a minute. So protease gene therapy, there's two types, germline therapy and somatic cell. So germline therapy, this involves altering the alleles in the sex cells, so in the sperm or the egg cells before fertilization, and that's really important, it's before fertilization. This will mean that every cell in the resulting offspring will function normally. This means basically that every cell of any offspring produced from these cells will be affected by the gene therapy, and they won't suffer from the disease. Currently, germline therapy in humans is currently illegal, and it's because this method is very controversial and we're not allowed to use it on humans. That's why it's never been used in humans. But some studies have been conducted on mice, so it's a big ethical issue. Some people are really worried about the ways we could then use this technology for things other than medical treatment, because if we could do this to the egg and the sperm cell for medical treatment, surely we could do all these other things, such as for treating the cosmetic effects of aging or changing how a baby would look to get to design a baby. And other people worry that there's potential to do more harm than good by using this technology. So we could actually maybe overexpress some of the genes. Genes produce too much of missing protein. So there's lots of issues surrounding that. We've also got somatic cell therapy. This is some that does exist and is being used slowly. Somatic cell therapy involves altering the alleles in body cells, so in adult body cells. In particular, the cells that are most affected by the disorder. Every cell in the body contains a defective allele, but only the affected area is targeted. So for example, if a lung is affected by cystic fibrosis, a vector is needed to carry the normal alleles to the target cells, so to the lung cells. The theory of it is that it works because we don't need all those other cells being changed, because actually the effects of cystic fibrosis I know cystic fibrosis can affect other parts of the body other than just the lungs, but for example, the hair, we don't need that being changed there. So that's sort of the idea. So the somatic therapy that's happening here in this image, for cystic fibrosis targets the epithelial cells because lining the lungs. And some somatic therapy doesn't affect this, and somatic therapy doesn't affect the individual sex cells, both. So any offspring could still inherit the disease from that individual. So gene therapy is the last little bit that we're just going to look at, and we're going to look at some of those vectors that we could use for somatic cell therapy. So we could use liposomes. As we've said, they are minute lipid spheres which can fuse with the phospholipid bilayer of the cell membrane to release their contents. And what we basically do with them is we insert the healthy gene into a plasmid in the liposome, which is then wrapped in liposome. The gene is released into the cell and moves into a nucleus when it's expressed. Or we could use a viral vector. So we use retroviruses and adenoviruses as vectors, and the genetic material is removed and the new gene is inserted into the viral coat. The virus enters the cell, surrounded by a vesicle, breaks down, releases the DNA, which is then transported into the nucleus. In cystic fibrosis patients, the virus is sprayed into the lungs. People going through any defective genes and gene therapy go through something called genetic counselling. That might be newly diagnosed patients, new parents, or couples planning a pregnancy, or relatives of a patient concerned that they may also carry a disorder. And genetic counsellors help people and their families faced with a diagnosis of genetic disease, and they can help them to understand both the factual information about the disease and the effect it will have on their lives so that they can reach their own decisions about the future. So that is it for this topic. And the last little bit of this topic, which is topic 8B, is just going to be the last session on gene probes and medical diagnostics. And the end of that session will also have some exam questions on it.